Good day, this is Professor Will, CBMG PhD. And today's Friday, November 5th, 2021. It is 4:07 and 7 seconds, p.m. Eastern Standard Time. Today's research, RAS inhibitors. So let's hop to it here. RAS inhibitors for use in fighting more cancers. Date November 4, 2021. Source, UT Southwestern Medical Center. The new findings help better understand how one of the most commonly mutated genetic drivers of cancer passes signals that cause the disease. Researchers focus on a family of proteins called RAS, which is mutated in 20 to 25% of all cancers, especially in lethal cancers, such as pancreatic, colorectal, and lung cancers. A framework to develop RAS inhibitor strategies is badly needed because recently approved RAS inhibitors, such as sotorasib, only work against one specific mutation, and many other RAS mutations also cause cancer. And this work sets the stage for development of new targeted RAS inhibitors to address major drivers of lethal cancers, such as pancreatic and colon cancer. The researchers developed drugs that bind to a specific RAS mutant, where a glycine amino acid at position 12 in the RAS protein is changed to a cysteine, the so-called KRAS G12C. And cysteine is a distinctive amino acid that allows us to irreversibly attach drugs using special chemistries. Other major cancer-associated RAS mutations do not give us the same foothold. In the latest study, the research laboratory sought to understand how cancer-causing RAS mutations pass inappropriate signals from the surface of the cell to the cell nucleus. The formation of large protein clusters as part of the mechanism was known, but the cluster structure was unknown. The research collaborators used computer simulations to arrive at an atomistic structural model of a RAS assembly and validated the model using biological systems. And this structural model is now available to the wider RAS research community. Researchers hope it will enable researchers to test new ideas about how RAS works in normal physiology and new strategies for targeting cancer-causing RAS mutations. Because RAS signaling relies on formation of RAS complexes, researchers think it may be possible to create new generations of RAS-targeted drugs that work by breaking apart such RAS complexes. Good day, this is Professor Will, CBMG PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.